to wonder how that turns into this, despite having the same genes. How are all these types of cells built from the same code? Magic! The answer lies in epigenetics, evolution's gift that gives code personality. Here is the genetic code. It likes creating copies of itself and taking over the planet. To do this, it comes up with neat tricks, such as employing less motivated molecules to build and maintain closed environments, providing appropriate conditions and raw materials for its replication. The best genetic code has a fashion sense. Just as we cover and uncover parts of our body to the weather, the genetic code can hide and unhide parts of its information in response to the external environment. Snowshoe hares, the code for white coat, is hidden till winter. The genes for these beautiful wings are hidden till the caterpillar reaches a particular age. Each of our cells contains code for all types of cells, but the code for neurons is hidden from our BCs. Epigenetic change changes phenotype without affecting the genotype. How is it done? Changes in structure of histone proteins around which our genetic code lives cause them to clump together, hiding some parts of the code from the translating machinery outside. Addition of methyl groups to specific parts of the genome called CPG islands essentially comments parts of the code ahead by making it non-interactive with translation molecules. The process is controlled by regulatory genes which encode RNA that selects certain groups of modifying agents according to signals from master regulatory proteins encoded by master regulatory genes. I've been speaking from a gene's point of view to convey that unlike popular notion that we can silence our genes using mental energy, most of epigenetics is predetermined by our genetics. Now that we have the required tools for thought, let's explore how a ball of identical cells forms you and me. The zygote first divides into a number of all-forming or totipotent stem cells. According to the positions in the ball, they further divide into multiforming or pluripotent stem cells. Pluripotent cells give rise to cell lineages by progressively covering more and more parts of the genome until only specific professional instructions remain. Here's an example. Interestingly, we can also go in the reverse direction. I can take a mature neuron and slowly remove epigenetic marks from it to give back a totipotent stem cell. This has huge applications. These cells can be used to grow hearts, kidneys, everything in lab beakers. In fact, you might already consume one such product, lab-grown meat. Meddling with cell epigenetics changes the whole identity of that cell. Change in cell identity is also the hallmark of a terrible disease, cancer. Although normally associated with mutation, epimutation or improper silencing of crucial functions is as important, if not more, to explain the workings of cancer. Epimutations are 1,000,000 times more likely than mutation. Understanding epigenetics promises crucial breakthroughs in life sciences, technology and medicine. It is the path to the eventual cure of cancer.